screen?
Yes, we can see that. Perfect.
Okay.
Thank you.
All right, so here we go. And that's my email, which you should all have one way or another, but feel free to find me. So in the beginning, there was Mendel. And then there was Dr. McCusick. This is his official portrait at Hopkins, and I love it because it's got all the MIM books that existed before Omen went online, and heritable disorders of connective tissue, and it shows the man's work. The photo, or it's actually a photo behind him, is known as the Amish Madonna. It's an Amish woman whose son has Ellis von Creveld syndrome, and she's showing his 6 fingers. Okay, so Mendelian Inheritance in Man started out as a bunch of notebooks and then turned into a book in 1966 and was published in 12 editions. The last one in 1998. That one had 3 volumes, and it was 8,400. It had 8,400 entries, and that's on onion skin paper. So today we have over 26,000 entries. So we can't publish this book. Just for historical context, the book in 1966 had 1,400 entries and no genes, right? It was all phenotypes. And actually, it was a catalog of autosomal dominant, autosomal recessive, and X-linked phenotypes. Okay, so this is a couple of pictures of the history. So initially, Omen was part of something called the Information Retrieval Experiment that was NCBI developed. And so it was always on computer cards, even in 1966, which is actually why all the entire first edition is all published in uppercase, because there was no ability to do upper and lowercase typing on computer cards in 1966. And that's why the titles are still all uppercase, and people hate that. However, I always know the source of that title. If it's all uppercase, it comes from Omen. So anyway, in 1995, Omen was moved to the World Wide Web by NCBI and incorporated into the Entree suite of databases, and it remained there until 2011 when omen.org was established. And omen.org is where you guys should be spending your time. The other thing I want to let you know is that Omen, which is a funny name, like who cares that it's online, Omen was one of the very, very first resources to be available online. And it was actually available for dial-up starting in 1987. So 10 years before the last edition and before Google and the internet. I mean, World Wide Web, sorry. Couldn't be before the internet. Okay, so that's the homepage. You're all familiar with it. There are video tutorials available from the homepage on effective means of searching, on mimatch. I'm going to tell you a little bit more about mimatch in a minute, and also on Gene Scout, which I'll also tell you about in a minute. But they're each, the longest one is 4.5 minutes. They're not long, and they're I think useful, but of course I'm biased. Okay, oh, there they are. How to search mimatch, and off this page is Gene Scout. Okay, if you see a problem, an error, have a question, whatever, that contact us button is on the top of every page in omen.org. If you're in any page, in any entry, if you see something that you think is wrong, if you read a paper that you think we need to add, anything like that, just open up the contact us and you'll get this page and you fill in your information. You can do a website technical issue or report a correction to content or suggest a reference. Or something else, and we will we answer you? Not necessarily, but we will see what you've said and we will address it. Meaning we'll either change something, fix something, or say that's interesting, but no, it doesn't have to be an Omen. But we won't necessarily answer you because that we don't have time for. Okay. So this is the information. I just want to make this small. There we go. This is the information content of Omen. So entries in Omen are either gene entries or phenotype entries. Within a gene entry, that's where allelic variants will live, okay? And again, you all know Omen is not the variant repository, but we have example variants for different variants in a gene that cause either different conditions, different inheritance patterns, something else like that. And I'll tell you more about that in a minute. And then there are external links from a gene entry. Any external link from a gene entry takes you to the relevant topical page of the other resource. If there isn't such a thing, there will not be a link, okay? The phenotype entries will have a clinical synopsis, and the vast majority of them do. And then they have external links that, again, are topical to the relevant resource, to the right page and in the other resource. Phenotype and gene entries are mapped together on the gene map. There's a dash line here because not every phenotype we know the gene of, okay, so that it's not 100%. And then entries in similar or identical, depending on your point of view, phenotypes caused by different genes will be organized by us, not computationally, into phenotypic series. Okay, so that's the data structure within Omen. Okay. Sorry, wrong. You won't let me go forward. Why? Okay, sorry. So Omen entries, each entry has a unique stable MIM number, and the MIM numbers are heavily used in the literature as cross-links with other databases. They have a primary title, and then they have other aliases and symbols and other things. And they may also have formerlies and so on. The MIM entry includes rich full text descriptions of genes and genetic phenotypes in a structured format. For example, there's certain headings that can be used. All headings that can be used won't be in every entry, but we have the upper level headings are fixed and structured. It's curated, similar to everything you guys are doing for ClinGen, and it has a predictable semantic structure. And everything that goes into Omen is based on something published in the peer review literature. Okay. It's a tiny group of people. Above this little text in the bottom are the human beings who work on Omen. That's it. That's all. There ain't no more. Kelly Preslepa is highlighted. She's a wonderful geneticist who does work on restructuring or updating or new clinical synopses as new phenotypes are added. Okay. This is a graph that we maintain that shows the growth of gene phenotype relationships. This is through the end of last year. As of today, if we drew it today, it would end at about just shy of 7,000, and this would be 4,500. So we're continuing to make progress. There's no slowing down in this growth of our knowledge of Mendelian disease. Okay, so I'm back at omen.org, and I'm going to type something in the on the search bar right here, which I'm sure you guys do every single day. At the moment, as of yesterday, I finished this last night, there were over 16,500 entries, including entries for protein coding genes, transcription factors, microRNAs, long non-coding RNAs, and locus control regions. Now, we don't have everything that's in RefSeq, right? So the genes in Omen are genes that we know something about their function, and obviously prioritizing those that are implicated in disease. Okay, so we don't have them all. These are Omen genes. There are over 4,740 gene entries that have allelic variants, and they comprise about 31,000 allelic variants because, again, those are exemplars. So now this is an anatomy of a gene entry, and I picked FBN1 for today. So the first thing is at the top line, there's a stable MIM number, 134797 is the MIM number for fibrillin 1. The asterisk before that number means this describes a gene. There's alternative symbols. There's an HGNC approved symbol. Almost always, not always, almost always, the Omen gene symbol is the same as the HGNC, but if for any reason it isn't, you can go right to it, and that takes you actually to HGNC's website. The cytogenetic location, and then the genomic coordinates, which we derive from NCBI, and we show it on build 38. So those are both important things for people to know. The top line is a gene phenotype relationship. These are the various phenotypes associated with this gene. If we click here where it says clinical synopses, what you will see is the clinical synopses of the different phenotypes associated with this gene in a table format. And there's a few things I want you to see here. First of all, these are toggled open a priori, okay? And this one you can see on one page, but you can imagine that there's certain phenotypic series that have, I don't know, 90 components. So you can close some of these if you don't want to see them all. So this is a toggle you can use. Then it goes from, it gives you the number, the name of the phenotype, the name of the gene is right below it, the molecular basis and a link to the allelic variants is at the very bottom of this list. And then it goes from there on down, inheritance, growth, head and neck, and so on, with the upper level entries that are fixed and, if present, included in a clinical synopsis in Omen. And you can look side by side and see if they're similar or different. The other thing that's over here that you can see is you can toggle some of these. If you like, let's say you want to compare something and you really want to look at heart, and growth takes up too much space, or you just don't care. You can toggle this closed right here. Okay, that's one thing. And the other thing you can do is you can see how many of these 8 clinical synopses have an inheritance or have some comment in growth or some comment in head and neck and so on. Okay. All right, so now I'm back to fibrillin 1. And the next thing I want to show you, I think, is the table view of the allelic variant. So you can, if you click here, you get the text of how we are describing that allelic variant. If you click here, you get the table view. And the table view shows you a few things. Number one, we are 70 examples. If you click this thing where it says selected examples, it explains to you how we select which ones we show. It shows you the MIM number, and you can sort these, by the way, the phenotype associated with that variant, the mutation, and the mutation is in MIM format, which is not up to date with HGBS, but we cannot be changing these variant. And we just, such a tiny staff, we got too much work to do. So if you want to see every correct possible HGBS nomenclature for your variant, for that variant, you click here, it'll take you, and I don't have it here to click onto, but it takes you to an ensemble page that shows literally every HGBS permutation of correct naming for that variant. If it's in Nomad, it'll be in the next column here. And the ClinVar, everything added to Omen, let's do English. Every variant added to Omen is uploaded to ClinVar every night. Okay, now 70 seems like a small number, so you can click here to see what all the ClinVar variants are. And if you do that, you go to ClinVar, and I went to page 2 just because I wanted to avoid the copy number variations. And you can see there are 4,804 variants in FBN1 in ClinVar, and that 1,298 of them are pathogenic, and over 1,000 are likely pathogenic. So, Omen is an exemplar place. It's not the place to go for variants. I know you all know that. And then this just shows you what's available on this page, and you all know everything about going from here forward. Okay, now the other thing I wanted to show you guys is PhenGene graphics. I don't know if any of you are familiar with this. And by the way, feel free to interrupt me, like either by shouting something out, or if somebody's writing something in the chat, let me know what it says. So if you want to know what a PhenGene graphic is, you can click this little question mark, and it takes you to this. This is a PDF that explains what everything is in here. So this particular one uses orofacial cleft due to MSX1, divides into the gene. There's an orofacial cleft phenotypic series. Here's all the components. Here's the gene. This gene is involved in other things, and so on. And it tells you what can open and close things and how to do it. I'm not going to go over it. You can do this yourselves. So in this case, I'm showing you the linear view of FBN1. So here's the gene. Here's the phenotypes that are associated with it. Here's the fact that wild Marcasani is part of a phenotypic series. And if you click on this, it'll take you to the phenotypic series. If you click on this, it takes you into the actual entry. So here's the other components of the wild Marcasani phenotypic series. I'll tell you what that question mark means in a moment. And here's geophysic dysplasia, and here's the other components of that. Okay. Now we also have this, and this is built as you click it. It's built on the fly from the data in Omen that moment. Okay. So tomorrow you might get a different thing if we added something to this. Okay. You can also see this as a radial view, and to do that, it shows you FBN1 here. It shows you all its phenotypes. It shows you, and this overlaps, but you can rotate this. I didn't do it as a video, but you can pick any of these dots and it'll rotate it. So this one is a radial view. By the way, the linear view denotes no hierarchy. Those are just relationships in a way that can be drawn. Okay. Next thing. So now I'm going into the anatomy of a phenotype entry. And here we are in geophysic dysplasia. So this has the MIM number, which again is stable. This pound sign or number sign in front of this entry means this describes a phenotype whose molecular basis is known. That's what a number sign means. There's a PhenGene relationship table, if there is one. If there is no PhenGene relationship, there's no table, obviously. And then you can open clinical synopses, phenotypic series, or PhenGene graphics. And I'm going to show you about clinical synopses and phenotypic series in a moment. So I'm not going to open it from here. The other thing I wanted to show you here is that you can either, if you click here, it opens a new page. If you click here, it opens it in series. Same thing for all these arrows. It's an arrow. Okay. So, all right. So back to Omen, and the statistics page shows you how we're doing every day. You can watch our progress. I'm going to go back a minute and tell you one other thing. The top line of that is entry updates. So you can, an update log, so you can see what we put in overnight, etc. The The next one gives you entry statistics, and it doesn't look like this exactly. I just wanted to make it easier for people to read, but these are the number of entries that we have in Omen as of last night. So it was updated on the 24th because it updates overnight. And these are divided into types of entries, and then whether they're autosomal, X-linked, Y-linked, or mitochondrial, and the total. So it was 26, which is kind of a nice round number, a couple days ago. Okay. Now, the morbid map scorecard is that I showed you that graph of the growth of gene-phen relationships. So this is where we track that daily. So the number of phenotypes, which can include different kinds of phenotypes, not just single gene disorders, for which a molecular basis is known, is very close to 7,000. And the number of genes with phenotype causing mutations was 4,505 as of 2 days ago. Okay. Then we divided those phenotypes more finely, so that people would have no ambiguity about what we were including here. And so there's single gene disorders and traits, and by traits that could be something that's, let's say, eye color, hair color. It could be rolling your tongue, things that aren't disorders at all. And then susceptibility to complex disease or infection would include like, so BRC, breast and ovarian cancer due to BRCA1 or BRCA2 is considered as all cancer things are considered susceptibilities because, you need a second hit. And also things like herpes simplex encephalitis. So it's a recessive susceptibility. If you don't have the misfortune of being exposed to herpes, you aren't going to get the disease. Okay. Then non-diseases are like QTLs, platelet size, red cell size, stuff like that. And then somatic cell genetic disease. So Omen is not cosmic. It is not where to go for somatic cell cancers. There are we do put mosaic conditions in this category. And then there are some variants in some cancers where you really can't talk about biology without having them in. So for instance, we will have the V600E BRAF mutation in because it's so important to so many different things. So that will be in. But it's not, we are not trying to catalog that at all. Okay, so what kind of phenotypes do we have in Omen? Obviously, single gene disorders, that's what it was developed for. Phenotypic traits, susceptibility to drug reactions, including malignant hyperthermia, and then actual drug sensitivities. Also says susceptibility or reaction to infection, germline susceptibility to cancers, and then recurrent deletions or duplication syndromes. So we are not going to have the one-offs. Please go to Decipher, please go, elsewhere to see that, but we will have recurrent syndromes, and particularly ones that were recognized as a syndrome before the fact that it was a CNV was recognized. Those were in Omen a priori, so we have to give the cause. Okay, next slide. The challenges in defining a phenotype. So there are many, and I'm sure you've encountered most of them along the way in your curation work. So there are changes in diagnostic modalities. So for instance, I don't think that Loeys-Dietz syndrome could have been recognized as a separate condition until we had the ability to do CT or MR angiography and see all these tortuous vessels. So, you have to be able to recognize something and see, oh, this is the problem and it's recurrent to do that. There are changes in diagnostic criteria, differences in medical care and medical intervention. So there's cohort effects over years or decades, cultural considerations. The biggest one, I think, is medical subspecialty bias. So if you are an ophthalmologist, you know everything there is to know about the eye, but you may not notice that your patient is missing a leg because that's not what they came to you for. And so there are many conditions described by different specialists that turn out to be the same thing, but it isn't clear from the original paper. So it can be really, really challenging. Ascertainment of the phenotype at different stages of development. Obviously, if you're reporting, several newborns, you have no idea what they're going to look like at 5 or 10. And then somebody reports a group of patients who are 10 years old, and it isn't clear that they have the same exact condition that these newborns had because they don't look the same anymore. They're not acting the same. They don't have the same set of problems. phenotypic diversity at a locus, and genetic heterogeneity. All right, enough challenges. So what do we do with all that? We lump and split phenotypes. So Omen, since the early 90s, has divided phenotypes by the molecular basis. So, and the discussion of that genetic heterogeneity is always included in the lowest number of the series. So this is an example of Sickle syndrome. And there are 9, as of yesterday, there are still 9 Sickle syndromes. And this list shows the phenotype and the gene. These are live links. And a question mark I'm going to talk about in a minute, it described one family only. So it went in with a question mark, which is like hyper limited. We don't do the definitive, strong, moderate, etc. But one family gets a question mark, and I'll explain all that in a second. And by the way, in this case, every one of these has a known gene, but there are phenotypic series where there are loci that were previously mapped where the gene is not known. Then in that case, you'll know that because the MIM number is the same for the phenotype and the gene. Otherwise, they would be different. Okay, moving along here. So this is an example. I'm sorry, I don't know if I circled it, but let me go back one. Yeah, so I viewed the corresponding clinical synopses as a table. So this is another way to see them all together for all the Sickle syndromes. And you can line them up and see how they're different or the same. Very useful information. Okay, so currently we have 489 phenotypic series that comprise over 4,100 different disorders. And a disorder can live in more than one phenotypic series depending on how they're structured. Okay. I'm hearing no questions. Does anyone have any before I move on to curating gene phenotype relationships?
Hi, yeah, this is Deb. I have a quick question. So I always have a lot of questions, so don't want to hold it up. So are there genes curated like through the ClinGen G-sets that are not in Omen? So what I struggle with is trying to figure out kind of the Omen set versus the set of like other curation sets and like a quick comparison. So
there shouldn't be.
Okay.
There shouldn't be. If you are actually doing a ClinGen curation on a gene not in Omen, please hit contact us and say, hey guys, this is associated with the phenotype. What's wrong with y'all?
Okay. Because sometimes the curations that we do are, limited, right? And so they're like, I could see that some of these limited ones wouldn't be an OMIM, and then you're saying then they shouldn't be an OMIM, correct? So only things that have strong or definitive should be an OMIM? Or?
No.
Okay.
OMIM should have more. So OMIM is not a, okay, I don't know quite how, so clinical people use OMIM all the time, but it's really not like clinical grade, right? It's a research resource. So we should be curating a limited sooner than you are, in my opinion.
Okay.
So it's possible that we've read the same paper and we didn't make a disease gene relationship because we didn't believe the paper at all. Okay. But that gene should be in, and that if we read that paper, it should be in under molecular genetics, or maybe we will have made a variant of uncertain significance to discuss it. And I'm going to show you that right now.
Okay.
But in theory, anything you're creating, any gene you're creating, we may not have the phenotype because we didn't believe it, but any gene you're curating should be an OMIM. If it's not an OMIM, please send us an email.
Okay. All right. Thank you.
Sure. And somebody just put something else in the chat. All right, Gram 1. Okay. Gram 1 needs to be looked at. I will look at that after this, if that's okay. Okay.
One quick question, Ada, sorry. How are risk factor variants handled?
So, that's a big question. It depends on what you mean. Tell me what you mean by risk factor variant, and then I will try to answer the question.
Dimitri, would you like to expand on your question?
Yeah, I'm going to try. There is a repairs upstairs, so it may be a lot of noise, but I'll try. So, for example, there are some variants related to cancer predisposition, and they have a frequency around 1% in population.
So the variant will probably not be an OMIM. Again, particularly, I mean, it depends. So it really depends on the situation. So if it's a common variant in a known susceptibility gene, and we have already curated that. So it's in BRCA1 or BRCA2 or CHEK1 or whatever, or CHEK2, whatever your PAL-B2, whatever, then it may be in there. And certainly the fact that that gene is a cancer susceptibility gene will be in Omen, and all of them are dealt with as susceptibilities, and then the variant just goes in. The name of the disease is disease, susceptibility to, right? Whether the variant will go in or not depends very much. And again, we're not trying to put every variant in. So does that make sense? Did that answer the question?
Yeah, as you mentioned, it's sort of a complex issue.
Yeah, it's unlike the beautiful, clear SOPs that all of you operate under. In Omen, everything is and has always been a case by case. And the nice thing about it is it has tremendous flexibility, right? We can do whatever we need to do. Somebody describes something no one, like a long non-coding RNA, something no one's ever heard of before, we can put it in. On the other hand, the decisions that are made, there's a method to the madness, but it isn't your very, very strict SOP. All right, so here's what we do to curate gene phenotype relationships. So we create a clear-cut gene phenotype relationship. It's not definitive, strong, moderate, limited. We don't say any of those things, but we say this is a gene phenotype relationship. If there's two or more families with mutations in the same gene supported by segregation, or in the case of de novo variants, a statistical test of the likelihood. And I would say for de novo variants, it's definitely more than two families, more than two cases. And the other thing is that's increasingly an issue is the specificity or identification of the phenotype. So intellectual disability, a whole bunch of people with a variant in it. There's nothing else going on. There's no functional evidence. I'm not sure that'll get in anymore. But these are the rules that in theory we're following. Functional analysis and/or an animal model is a plus. We'd certainly add it into what we're putting in, but it's not required to make that relationship. And then we're going to put a number sign in front of the phenotype number. It's going to be added to the gene map. And why are we not moving forward? Okay. So that's what we do for that. A provisional gene phenotype relationship is made for a single multiplex family, or very rarely a single patient where it's an enzyme deficiency and the assay of the activity is like an irrefutable thing, and that it's already about something that's known. Let's say a CDG in one person, we might put that in, okay? We need functional data from an experiment or an animal model or both. We will put a number before the MIM side, but we add a note in the first sentence that it's one family. The allelic variant title has one family, and there's a question mark on the gene map. And that's what this looks like. So we've made it an immunodeficiency 16. It has a question mark in front of it here. It says one such family has been reported here. It has in the gene, it says a question mark in front of immunodeficiency 16. The allelic variant says one family. And basically what this means is caveat emptor, okay? Like it's one family, guys, don't hang your life on it. And it would absolutely go in as a limited in ClinGen, although you would spend a lot of time proving it in other ways. This is what it looks like on the gene map. So here's the location, the gene, the gene name, gene locus, the disease, and over here under comments, it says mutation identified in one family. Okay. So variants of unknown significance, which is not the same as a variant of uncertain significance. Okay, so a variant of unknown significance is basically a variant in a GUS. Okay, so we are not looking for these. If we read a paper, it's a single family, there's no functional data, but we read the paper and we spent the hour reading the paper and thinking about it, then we may add the variant as a variant of unknown significance, and we'll discuss everything about the phenotype in that allelic variant. We will not make a gene phenotype relationship, and we won't add it to the gene map. So that we might make a variant of unknown significance. We also might just put it in molecular genetics. So this is an example of two of these. This is a variant of unknown significance for, I can't tell you the name of the gene from here. And this is another one with MED20. This one's MED20, and this one is GRP161, sorry. Okay, so at the moment, Omen has something over 31,000 allelic variants. Allelic variants are in the gene entries, and we are putting in the first few mutations to be discovered, ones with high population frequency. a distinctive phenotype. So, you know, this variant causes Marfan syndrome and this variant causes Weill-Marchesani syndrome. Those would obviously, examples of both would be in. Variants of historic significance that people have been talking about forever, or whatever the reason is, unusual mechanism of mutation or unusual mechanism of disease causation, and then distinctive inheritance. So we will put in recessive and dominant variants so that people can see what that range is like. Okay, so now sometimes we need to reclassify variants. Sometimes people write a paper and say, you know, this is wrong. In fact, this patient has a mutation in this gene rather than that gene. And so that was never right to begin with. Sometimes they're eliminated from population frequency. And that is the one example where we always try to cite a paper for everything we put into Omen. But if there isn't yet a paper saying here, these variants aren't legitimate because their population frequency is much too high in Nomad to possibly account for the phenotype, then we would say that, and we would cite Nomad and the date and the frequency. And remember that Omen is a database that has existed since 1966, right? So it's got the latest and the greatest new stuff, but it also has old stuff. And you know, you guys are struggling under the burden of a few years, right, of having ClinGen. Just wait till you've got 50. It's not pretty. Okay, now clinical synopses. This is just a particular entry, Nestor-Guillermo-Progeria syndrome. Here's the clinical synopsis. I popped it up as another thing. This is the organization. In this case, and this is not in the right place, this should be under micrognathia. I don't know why it's down here instead of up here, but if you click on this little, I guess it's a bust, it will open this picture, and this picture is of a person with micrognathia. It's of the feature, not of the disease. And then if you click here, it comes from Elements of Morphology, and you can go into Elements of Morphology to find out the definition, other photos, synonyms, etc. Okay, so that's the full view format. The other thing is when you open this, you can, if you click at the top of the line, it says feature IDs. If you click that open, it shows you all the different all the different IDs for this particular feature from different resources. So SNOMED, ICD-10, HPO, UMLS, etc. Okay, they're all there. And of course, you can also all get this from the back end of the API of Omen, which is actually Omen, the website, sits on top of Omen's API. Okay, so this is another thing that you can do in this case. I have typed in progeria, and I've gotten 27 responses, and the you can see here that I've got both phenotypes and genes. If you go down this list, if I click on clinical synopsis on the upper right, I can get what's known as the clinical synopsis quick view format. Okay, so I can mouse over these green, where's my mouse? Here we go. I can mouse over these green things. In this case, I've moused over head and neck, and I can see the features under head and neck for that phenotype. Okay, so that's one way to look at things, very helpful in clinic. And the other way to look at things is this way. So again, I've done that limitation to clinical synopses because you can see the green here, and I can put check boxes and then hit compare selected, and I get these side-by-side clinical synopses. Okay. All righty. Now I'm going to shift to external links. So any questions about what I just showed you before I move on?
I have a quick question that says, so you were saying if there's a question mark in front of the phenotype in the phenotype table, then
Yeah.
that means that it is like based off of limited, not the gene, not ClinGen definition of limited, but like one family.
It's usually, yes, it means one
Okay.
family.
Okay,
Yeah.
great. Thanks.
Yeah. So it's the ultimate in limited.
Yeah. Okay, great. Thank you.
Yeah. Okay. So, so external links in MIM entries can be, these are just examples of what may exist. Obviously, again, there's only going to be a link to this resource if that is useful, if it takes you to a page in that resource that it relates to whatever you're looking for. So genome links, DNA links, protein links, gene information, all kinds of gene information. The clinical resources include, so this is variation, I'm sorry. So in variation, these are all the things that we have right now. And the locus specific databases will take you to a whole list of them, and you click on the one you want, because sometimes it takes you to a dead link. So we decided it was easier to go to the homepage for that. And then the clinical resources, the ClinGen dosage, ClinGen validity, if it exists for that gene, clinicaltrials.gov, et cetera. So all of these, again, take you to that page and the other resource if it exists. If it doesn't exist, the link will not be there. Okay. And we toggle open to, in a phenotype entry, we toggle open to clinical resources, and in a gene entry, we toggle open to variation. We just do that to make sure people understand that these arrows have something beneath them so that you can open it. It's just to make sure people know that. Is there another question? There's something in the chat.
Yes, we have a few questions actually. So one is, does Omen keep track of only the morbid genes, and is there a way to directly download those genes and their genomic coordinates?
There are a couple, no, we keep track of a lot. We keep track of, we for sure keep track of morbid genes, except I guess I have to look at GREM1. And then we keep track of other genes whose function is known. We don't necessarily we don't have all genes, because some genes have no known function and aren't implicated in disease, and we're not going to make a shell entry. We're not looking to do that, okay? And I can show you a couple of things of how to look at that in a moment. Are there other questions?
Yes, I have another bunch of questions here, I think from Deb.
Well, again, I think we can have it, go through the rest of the presentation, and if there's time, I'd ask some. Like I said, I have a lot. So
well, let me go on then and show you. So this
Sure.
this is a genomic coordinate search. So you can type genomic coordinates into the gene map, and you get to the gene map from the home page. Does everybody know how to do that? It just right on the home page, it says gene map, click that, and then you have this page. You put in the coordinates, and you'll see the location, the genomic coordinates, which comes from, let me take a gene. Is it not? One of these must be a gene. Here you go. Here's a specific gene with a specific coordinate. Nope, that's a big thing. That's a locus. And they come from NCBI. And then in this case, I've already clicked this to show phenotype only entries. So I'm only showing you the entries on this part of chromosome 1 that have a phenotype associated with them. Okay. You can optimize your UCSC genome browser to show Omen allelic variants, ClinVar variants, et cetera. This is just an example of an optimization here. Now, when you do a search in Omen, if you type in, so here I typed in dwarfism, it will do a thesaurus analysis. The thesaurus is hand maintained by us, okay? And it has, so for in this case, it has dwarf and growth retardation and hyposomia and short stature, or you can add all if you want to expand your search beyond that. These are for other ways of saying the same thing and not exact synonyms. So for instance, gallstones and cholelithiasis, or first and first, or British spelling and English spelling, those things are already hard-coded. You don't have to worry about them at all. It's just other words that mean the same or slightly different things than what you might have searched on. Okay, so in this case, I've done a search on short stature, and I put quotes around it because that does a proximity search, and I put a Boolean and another web neck, okay? And then it gave me other options for those things that I can pick or not. And then I have received 42 responses to this query. And I see that it matched on both terms. And in this case, it will match on both terms because I put in an and. If I typed in short stature, webbed neck, no punctuation, no Boolean, no nothing, I would get an enormous number, but the thing I'm looking for would still be at the top of the list or near the top of the list. So you can definitely search more efficiently in Omen, but even a lousy search will get you what you're looking for. Okay. Now these just show you some features from a retrieval page. So in this case, again, I've done short stature and web neck. And now you don't even have to go into the entry if you want to see the phenotype gene relationship table. You can see the ICD codes, Orpha and Doe here. And I think I forgot to show you on that first one, I guess it was geophysic or whatever, that there's the ICD codes for the disease. and links. And this will again take you to the topical link for that page. So if you wanted to, you want to go into Omen because what you want to do is get to clinical trials, and you don't want to go and read anything. You just need to know if there's a clinical trial for this condition. You can click that and go to that page with that search. Okay. You can pull up the phenotypic series. If you pull it up from the retrieval, you don't have the view these as a table. You have to actually go into an entry and then into the phenotypic series and then click to see the table, because we just didn't, there wasn't enough real estate on the retrieval page to put that in. Okay. And then in this case, I want to, I don't know if you guys need to do something like this, but if you're looking for genes associated with a phenotype for some work you're doing. So you go to an advanced search of Omen. And by the way, this is how you get to the gene map right there. You go to an advanced search of Omen, and it takes you to this, a page that looks like this. In this case, I've typed in cataract, and I've typed it into all the possible phenotype entries. So that I put a check mark here, and there's a search button at the bottom, which I clicked. And I got back 632 entries that have cataract in them. Okay. And now I want to know how many genes are responsible for those 632 entries. So I click on the gene map table, and that takes me to this gene map, and there's 510 of those. So if you're writing a paper and you want to know how many genes are associated with cataract, that's how you figure it out from Omen. Not that it's the, source of truth for everything, but that's what you'd get from Omen. Okay. If you want to know what we're updating, if we put something new in, and in this case, I think the whole entry is new, but we would have a green bar next to the new information. And we leave that green bar up for about 3 months. So if there's a great big entry and we added something to that entry and you're like, what on earth did they add? It will be in that green, under that green bar, or by that green bar. I want to tell, do you guys know about mimatch? Have you all heard of mimatch? Yes, no, maybe?
I haven't, so I'd love to hear about it.
Here you go. So mimatch, I think, would be very useful for some of you all. Let me tell you what mimatch does. So you click on mimatch on the top, you create an account, you'll get an email confirmation, you'll log in and set your preferences. And you can receive emails about new gene phenotype relationships, or updates to phenotypic series, or you can follow particular genes or particular phenotypes that you want to know if we added anything to them. And you can also save your searches. So if you do a really bizarre, you're only interested in one very narrow thing, and you do a very strange search in Omen that is, how you get what you need, you can keep that and keep using it. So in this case, I've now logged in. And so I've said, notify me when new gene phenotype relationships are cataloged in Omen. So anything you've picked, you will never get more than one email a day. And if there's more than one thing that you're interested in, it will be included in that email. So for some reason, I'm following these genes. I want to know if it's updated. I want to share my interest in these genes. Okay. And so then I go to the phenotypic series, and I can be notified about a new phenotypic series if we add a new one in Omen, and I can also follow them. So I don't know, let's say you are responsible for Adams-Oliver syndrome. If you want to know if there's a new Adams-Oliver syndrome that we add, you can just to that phenotypic series, you can follow that phenotypic series, and you'll get an email if we add something. Okay. And then finally, you can say, I follow this, and I want in real time. If you're logged into mimatch, you can say, I want to be notified of an update. I want to share my interest. And there are 25 others interested in this entry. And you can actually see their emails and contact them if you want to. And we have nothing to do with it. That's after you've done that, that's on you. We have no part of that. Okay, I don't know if you all have heard of Gene Scout. We built this when the Miami, CNV and runs of homozygosity tool went private and started costing a lot of money. And so this is available at genescout.omen.org. There is a really good tutorial. Please take a look. It's not long and it's useful. So you put in whatever coordinates you want, and it could be like a whole long list. It can be from an Excel spreadsheet. It can be from a report from a lab. You have to designate which build, and it's going to carry it over to 38, because everything in Omen is reported on 38. Okay. And then you'll get a response out, and I'm not going to show you all that. You should look at the tutorial to see what that looks like. If you are comparing two siblings who, let's say, have the same phenotype and they have large runs of homozygosity, and you're trying to figure out, is there any gene in that interval? You can do that by just toggling that, adding another person. They can even be reports in two different builds. It's really quite useful. And you can look at the intersection, or if you have one affected sibling and one unaffected sibling, you can do the subtraction. Okay. And then when you get your response, you can limit it you can look at all genes. You can look at Omen disease genes. You can look at Omen disease genes with a particular form of inheritance. You can go and look at the clinical synopses, comparing them. It's actually a really useful tool, but I'm not gonna spend a lot of time on it. Okay, so we have an API. Omen has an API, supports requests. It will give you your answer in whatever language you'd like. And there's lots of online help documentation, which I understand people find very useful. I'm a front-end user because I am not a computer person. But this is the kind of an example of what a JSON format output would look like. And you can see that all the SNOMED and UMLS and HPO terms are all included in through the API. So it's a very robust way for computers to talk to each other and for do batch work. We get about 880 downloads in 2020. There are 10,500 registered API and download users, and we get over 120,000 requests per day through the API. Front-end users, there's a whole lot of you, and I think you guys are the heavy front-end users. So it's almost 2.8 million users a year. There were 84,600 people, or IP addresses. Okay, so maybe it's fewer than that, people with more than 100 return visits in a year. And there's 42,000 every week. So people live on Omen, I guess. We get we had more than 47 million page views last year, and the last several months it's more than 1 million a week. People see about close to 6 page views per session, and spend about 7.5 minutes every time they come for an Omen session. Jenny had asked me to discuss how we track our literature and so on. We have built something that's for internal consumption called LitTrack. And what I've done here is just pulled, so different senior personnel in Omen are responsible for reviewing particular journals or particular topics. And in this case, I'm responsible for pediatric research, pediatrics, and journal pediatrics. And you can see I'm a little behind. So I pulled a JPEDS one, and this is the most recent JPEDS issue. It has all the, it has the table of contents, and then it's got an annotation. So in this case, this drew my eye. And PubTator, which is an NCBI tool that looks for, if I click on this, it shows me that there's got a disease, a mutation, and a gene. These are the genes. These are the MIM genes, and these MIM genes all have AVs already. Okay. And so I learned that from clicking this. And then if I want to say something about it, I click on MIM annotations, and I can say, I want to add it to these MIM numbers. I can say, no, I looked at it. There's nothing that we need to add to OMIM here, so I'm not going to use it, and so on. So, and we have more than 150,000 articles from more than 1,900 journals in LitTrack as of this moment, or actually as of a little while ago, because those data are not updated. And I think that's it. So now I will take your questions. So I'm going to stop screen sharing so I can see you guys. And then if we want, I can go live to see if there's something else you want to see in real time. And we have, a whole 8 minutes. Let's go.
That was fantastic. Thank you so much. So I know we do have a few questions from Deb. So Deb, do you want to go ahead with your questions?
Sure. Also, I want to say I just signed up for the mimatch and got an account. So that's great. So I did it like in that time. I'm super excited about it. One question that I have just to start out with would be, kind of automating any of the work that ClinGen does with gene curation back into Omen, right? Like, so how does Omen not just link out to it, but use it? Does it help in the task of Omen?
So what we would like to do, and it's an ongoing process to make sure that the disease names, so we have links from genes to ClinGen gene validity.
Right.
We don't have them from the phenotypes because you guys are using Mondo, and it's not necessarily one-to-one. So that's the delay in doing that. In terms of using what you have, so let me tell you how we do things in Omen. So this is an old database and it's full of stuff, right? So the new stuff we put in, hopefully we're putting it in before you get to it. I don't know about Grim 1. I'm going to go look right now. But I mean, we should be putting in new disease gene things that we think are real quickly, right? Now, might there be a limited thing that you've looked at that we looked at and didn't want, or that we didn't look at, and we don't want to look at it because we're not going to add it, and I'll wait for more data, right? So that's in terms of new stuff. In terms of extant information, when we add something to Omen about something that's already got a good disease gene relationship, enough patients described that, you know, I mean, obviously it's a brand new gene from last year and now there's 50 people, that paper has got to go in, right? And we have to describe the phenotypic spectrum and the mutational spectrum. Again, we're not going to put all the mutations in, pathogenic variants. We're going to put in, you know, that there are a few more so people understand, or, you know, like if you only had 1 or 2 and now there's some missenses or some recurrence, we'll put a couple more in to give a range, a flavor of what can cause the disease. But we won't put in, when we, an additional paper, we ask, is anyone going to care about this paper in 5 years? If the answer to that question is no, we're not putting it in. There's too much work and too few people. So the thing that I would like to do from Omen is to make sure people know about ClinGen and all the wonderful tools you have and all the curations you have. And, you know, I mean, ClinGen's great. And I work on several committees. So, but I think they're different resources. I think the most important thing that we need to know at Omen is if there is something that you have that we're missing entirely, that the world, like, again, a very limited association in one highly consanguineous family with 10 different genes implicated, you know what? Get me another one, okay? I'm not gonna put it in Omen. It just muddies the waters. But something, if Grem 1 is I don't know the story for Grem 1, I'll go read it immediately, but you know, if that's something that we need to have in because, you know, it's not strong, but there's a couple and, you know, this seems to be an association, we need to put it in. But I mean, I can tell you, and I think you guys, I don't know if you've curated or not, but there's a gene TAUC1. There's 2 papers already. It's like 28 people. A little less than half of them don't have any intellectual disability. A little more than half of them do. The range is mild. It's maybe ADHD. There's really no other phenotype. Oh my God. I mean, it's not in yet. I just wrote it up. It'll show up in a week or so. But like, that gives me pain. Like, I don't know what to do with that. I think and I said, I want to make this an IDD susceptibility allele. And my team was like, what? I said, well, they don't all have IDD or developmental display. I don't know what to do with this. So, you know, so we all feel each other's pain. This is not easy. The nice definitives, they're great. Old genes, great. New genes, definite phenotype. There's 20 things wrong with you. Everybody looks the same. Wonderful. All the rest, it's getting harder and harder and harder.
I guess I would wonder if something like the gene you just talked about, like, could that go to the ClinGen Intellectual Disability Gene Curation Working Group? Sorry to that group for suggesting more work. And then they would say, you know what? This is limited data. Like, they do the curation on it and then send it back to you and confirm maybe what you're thinking on it or something, so that it would then go back in. But anyway, my ideal world would be to reduce the number of clicks that it takes to get curations done, and then also to find information about genes. So I feel like it's, you know, we go to so many different places to find information about genes when if that information is done in one area, if it can go into another area as well.
So first of all, and I forgot to show this to you, at the end of every paragraph in Omen, there's a little plus in a circle. It's called a reference plus icon. If you click that, it does a PubMed search of the keywords in the foregoing paragraph. It's organized by relevance, but you can switch the way the retrieval comes back by date. So that is a useful thing in Omen, and that, you know, also limits that we don't have to read every paper on every subject to know everything. I would love to have ClinGen links more prominently displayed, particularly on the gene phenotype table. But the trouble is that we've been working on this for a long time.
The phenotype, okay.
Phenotype definition. And that was just the source of a whole another conversation that we won't go into right now. But anyway, so I think I agree with you that fewer clicks is good. I do think the scope of work that Omen does is different than the scope of work that ClinGen does, which is different than the scope of work that other groups working on this do. And I think ClinGen, you have it's a very robust, detailed, painful, all my respects to all of you, curation that to establish a level of evidence of the gene association, and then another, different one for the variants. In Omen, we are booking through literature to put it in or not. It's a different, we're not, yes, we want to make sure we have all the genes and all the phenotypes, and hopefully we're not missing much, hopefully not much at all, but it's a different, you guys are curating to a clinical standard. It's a different scope of work. I'm trying to make sure that anything that anybody wants, so, millions of people use Omen, right? Some people land on it because they've done an experiment in the lab and they've done a pull down assay and they found an interacting protein. They know nothing about the gene. So you start at Omen and you read a little bit and you're like, oh, wait, that's interesting. Let me go do a PubMed search. Let me go do a whole bunch of other things. Or you go and you say, oh, all expressed in the eye? No, I'm looking at the liver. Go away, And so, people use Omen in a lot of different ways. And many people use Omen in many different ways. And because we're trying to make so many people happy, and there's so few people working on it, we need to be quite focused. In terms of links, I agree with you completely. In terms of, can we use what you do to do what we do? Hopefully, most of what you're looking at, we've already done. Okay. And if there's something big we're missing, like, hey, you missed the paper on 50 patients 2 years after this first one, send us, put this reference in, guys, don't do it, or Gram 1, I'm going to go do Gram 1 right now. But the rest of them, and the other thing you should know, if there's something that looks like, oh, why isn't this paper in Omen? I want you to know that it might be on Joanna Amberger's floor. Okay. There are many papers that we look at and we're like, oh, not ready for prime time. And it's waiting for the second paper. We know it's there. There's just not enough there to put it in. I don't know how much you guys have had to deal with this yet, but cleaning something you've already created is incredibly painful. Undoing something is dreadful, right? So we are much better off not adding something that isn't clear. And I don't mean strong or definitive. I mean, the paper is murky, like for instance, I just this morning before this, somebody sent me a paper that reported a gene-phen relationship in a family with 2 affected siblings with congenital hypoventilation syndrome, 2018. There's nothing since. The mutation is not clearly described in the paper. There's beautiful functional evidence. I said stuff at Mulgen. I don't even know how to give it a variant. I don't know what the variant is. Sorry. And Joanna sent them an email. Hey, what's the variant? No response. So, it's none of this is easy. I'm sure all of you have suffered to some extent. We've just been suffering longer than you have. But I totally agree. The more we can connect, the better off we all are. We're actually after 1, so I don't want to keep you. I'm sorry, I just realized I talked way too much. And I didn't answer most of your questions, Deb.
Oh, I'll always make more.
Does anybody else have any questions or things I didn't answer or left out that are terrible that I shouldn't have done?